Thank you so much for the kind introduction and great opportunity to present our ongoing development of new antibiotics derived from natural products. And I guess I take my pride in representing one of innovative small biotech companies in South Korea dedicated for this infectious diseases. I guess this AMR is a significant problem in the global regions, and we need more companies and innovative technologies to rise upon. So that basically summarizes my brief introduction. And not to not mention, we are in serious need of new antibiotics. So our company mission is to develop a new innovative antibiotics at the same time addressing more intractable bacterial diseases that I will discuss in a short period of time. So as mentioned throughout our presentation, AMR threats our global citizens, and in 2019, 1.3 million people have succumbed to this ongoing bacterial resistance problems, and it's estimated that in 2050, 10 million people will be affected by AMR. And coupled with this global climate crisis, I think the number will significantly arise, and therefore it is critical that we come up with a more innovative antibiotics at this stage. So our problem to solution is that we need new antibiotics that possess new mode of actions. So that will probably cover resistant emerging bacteria. Now, our company focuses on chronic intractable bacterial diseases that translates to low treatment success or high recurrences. So we need potent antibiotics that are potent, obviously, and have some sort of additional host-directed therapy, which is the ongoing, I think, a new platform of this new antibiotics. And obviously, we would like to be more patient compliance in a sense that we would like to reduce long treatment time or reduce systemic toxicity caused by current antibiotics. That will do. This is a brief introduction of our company. So our company was established 4 years ago, so it's quite young company. Our headquarters is actually located in Daegu. We may relocate, but I don't know. We have a biolab which covers some in vivo and in vitro analysis of bacterial infections. So we do have some CRO activities that are going on, and we have another office that is dedicated for preclinical and clinical development. So as I mentioned, we target this relatively hard intractable bacterial diseases. So you can see we target C. difficile, which causes intestinal damage. And we also target non-tuberculous mycobacteria. And another problematic Pseudomonas aeruginosa for chronic lung infections. And obviously this is like celebrity of AMR, right? So we target also Staphylococcus aureus as well. So we have came up with this novel antibiotics called thiopeptides. These are marine natural products, have very complex chemical structures and have not been exploited in much detail. But through my PhD and master's, we came up with a more reliable synthetic platform technology in which we can make a lot of useful lead intermediates with this marine natural products. So we also publish our findings in reputable journals, right? Why not, right? That will basically establish our scientific rigor in this field. So this is the structure of our compound. As you can see, this is micrococcin P2, which is, I told you about, this is a marine natural product. And as a chemist in the background, this is a very challenging molecule to synthesize. But we were able to synthesize this molecule by decakilogram quantities. So I think for CMC and production scales, I think we are in a great stage to progress this into our clinical stages, and that's exactly what we are doing at this stage. So our platform technology came in a sense that this red molecule shown in here is critical to possess antibacterial activity. If you alter any of functional groups in this red molecule, your antibacterial activity basically abolishes. So you do not want to touch this red portion, and this acts as a pharmacophore to inhibit bacterial protein synthesis. But however, if you you can certainly modify this blue portion of the molecule. So the problem with this molecule is that it has low aqueous solubility, meaning it has you will have difficult time in translating this molecule into clinics if you have low solubility. But since that you can modify this in a much reasonable sense, you can definitely increase aqueous solubility and relevant pharmacokinetic properties, and that's exactly what we did. So from our pre-lead optimization series, the aqueous solubility of our preclinical candidate has overcome such a low aqueous solubility by a factor of about 200,000 fold. So the things are, it's water soluble at this stage. So, this is a beauty about our platform technology in a sense that we make a large kilogram quantity of this macrocyclic fragment, and all you can do is do a Suzuki coupling towards the end of this molecule. So you do have a nice platform technology that will obviously be, mitigates production costs and stuff like that when you expand your pipelines. And our molecule actually binds to a specific region of bacterial ribosome. And most of the protein synthesis emitting antibiotics actually bind to this central region of ribosome. So aminoglycosides and macrolides, you actually do bind in this region, whereas our molecule binds to a little bit right tether of the ribosome. And this 26-member macrocycle is actually the pharmacophore that binds very tightly to this L11 and 23 srRNA domain of bacterial ribosome. So obviously, since our target is far from marketed antibiotics, we do not have cross-resistance to currently available antibiotics, which is quite nice. So, that core intermediate that you saw was the macrocyclic fragment, and if you have 2 or 3 steps, you can come up with the various preclinical candidates. So AZ024 is targeting Clostridium difficile infection. We are at the IND enabling stages at this moment. We will enter phase 1 hopefully by next year, followed by other preclinical programs for NTM. There are largely two different species which we will target both of them. And our agent also has interesting activity against Pseudomonas aeruginosa, and obviously that's also another problematic pathogens that we just discussed. And we're also focusing on Staphylococcus aureus for topical indications. So we are trying our best to progress our program. So we have a lot of patents that are published. We are relying mostly on the government grants because it's really hard to raise money for biotech companies, especially for antibiotics in this field. But however, we did a pretty decent job at this stage, raising about $50 million at this stage. So I think we will be able to push the program for phase 1 and preclinical candidate for the Series A. And we are publishing quite a lot of the papers as well in this infectious diseases area. So I will now shift a little gears for the C. difficile infection. I guess many of you already know that the C. difficile causes significant problems to your intestines. In Caucasian population, this is more severe in a sense that if you contract C. difficile infection, 10% of patients actually die after 20 days of initial diagnosis. And this is classified as an urgent threat by the US CDC because this is an intractable bacterial diseases that causes high relapse and have low treatment success. And this is a very nasty pathogen in a sense that this forms spores. So this is a very resistant form of bacteria, and spores can reside in anywhere that we can see, like table surface and chairs and whatnot. So basically, what it does is that when we ingest somehow the spores, this gets activated through your digestive systems. So obviously the bacteria will wake up and form, and then find their nice space in intestines to awake. and that causes significant problems to intestines. So, megacolons and produces toxins and stuff like that. And for us, I guess we eat a lot of fermented food, right? Kimchi and stuff like that. So, this does not kill a lot of Asian populations, but, this is a the nasty infection in the sense that when you get contracted, you have a lot of diarrheas and high fevers. It's not pleasant. So in Korea, actually, the C. difficile infection is also rising significantly. Since 2007 to 2020, the incidence rate of C. difficile infection is significantly rising. I think there are about 30,000 patients that are hospitalized for this infection. So our target is to, beat the first line of treatment for this infection. Otherwise, you cannot, have a good chance in antibiotic field. So our aim is to target fidaxomicin, which was approved by FDA in 2011. So basically, what it did is that it beat vancomycin, the prior first line of treatment, a long time ago. The problem with vancomycin is that it has high recurrences because obviously when you ingest vancomycin as an oral therapy, that causes significant dysbiosis in your intestines. So meaning that it not only affects C. difficile, but also affects good bacteria in your intestines. So you don't want that. So fidaxomicin is more selective in a sense that it reduces the relapse rate from 30% to 20%, but it's not perfect. Based on a lot of surveillance data, it is shown that fidaxomicin is less effective against the hypervirulent strains of C. difficile. This is, another strain of C. difficile that produces a lot of toxins. So it's not effective against that. So what ANJ Science is trying to do is that maybe, beat fidaxomicin by targeting this hypervirulent strains. So to make the better story short, we want to increase treatment success, right? To be more potent against fidaxomicin and having a less impact on your gut microbiome as to reduce this recurrence rate and having more effective profile against hypervirulent strains. So then, you have better chance to, remove fidaxomicin and have our agent be prescribed as a first line of treatment. We have other competitor in the field, but since they're comparing against vancomycin for their clinical trials, I think the market wants a better antibiotics in this field. So this is the data that we have accumulated over the years. So, our agent has potent antibacterial activity against various types of ribotypes of C. difficile, and blue is denoted as the hypervirulent strain. So our agent is about 2 times more active than fidaxomicin, or 8 to 16 times more active than vancomycin. And this is well pronounced in these time-kill kinetic studies in a sense that when we treat our compound at the same concentration to fidaxomicin, our agent is rapidly bactericidal. So it takes about 8 hours to kill the bacteria, whereas fidaxomicin takes about 48 hours to completely, be cidal. So, our agent is rapidly bactericidal, and this is well translated in the post-antibiotic effect. And fidaxomicin prescribed twice daily, about 200 milligram. So since our agent is bactericidal, it takes about 22 hours to, bounce the bacteria to bounce back. Meaning, I think our agent may be prescribed once daily dosing regimen. And as I described earlier, patient compliance is probably maybe the key for putting the fidaxomicin over the other shelf, right? So I think that may will have a better chance to prove this in clinics. And this is our in vivo data. So what we have done here is that we disturb gut in mouse's gut through dextran sulfur sodium. So you kind of like damage intestine a little bit, and then you infect with the hypervirulent ribotype 027. And basically the treatment was done QD, as I prescribedly mentioned, and we observed mouse for about 20 days. AZ024 has overall survival rate about 80%, whereas the others have 40%. So if you see, vancomycin, fidaxomicin, you see some mice dies as it goes along. So, it means that recurrence had occurred. So our agent does not have recurrence against a hypervirulent strain. So I thought that's interesting. So we have now then, checked gut microbiome, how our agent shift the gut microbiome shift, right? So through 16S RNA sequencing, this is day 0, so no treatment has been taken. So you see this gut microbiome shift has not been significantly altered. But at day 21, you see that the shift of gut microbiome has occurred. And if you compare with our agent to control there, our agent overlaps very closely to control, far better than fidaxomicin and the other agent that is in phase 2. And we did not also generate a significant form of antimicrobial resistance genes, and I think that is another significant strength of our agent to push this program for clinical development. And this is another cluster that you can see, meaning if you see alteration of a lot of colors, that's bad. That means you disturb gut microbiome at a significant extent. So if you compare with our agent to control, color is well preserved, so that means you do not significantly alter your gut microbiome. And we have done extensive GLP toxicology of our studies, and basically we have no significant liabilities of our agent. Obviously, our agent is, when orally administered, it's not systemically absorbable. So we had to perform additional IV toxicology to, forcibly introduce our agent in blood plasma, and obviously we did not see significant liabilities. And the AUC of the highest dose versus highest dose at IV was very significant. So we do not expect any significant liabilities for upcoming phase 1. So that's pretty much it for the C. difficile, and I will now move on to lung infections. And as you probably be aware, bronchiectasis is another lung inflammations. Yeah. The incident rate in Korea is quite significant, and as you grow older, you have more likely to have this lung inflammation caused by bronchiectasis. And if you have bronchiectasis, it produces excessive mucus, yeah, and that obviously provides a good environment for bacteria to grow. And this is a recent data that if you have bronchiectasis, you will likely be affected by NTM species by about 60%. And also Pseudomonas aeruginosa becomes another problematic pathogens for it. And there are some cases that you do get comorbidity with NTM and Pseudomonas. So if you want to target these chronic lung infections, you may have, if you have agent that covers the broad range of this chronic lung infection pathogens, I think you are in a great game to, have a very good market penetration for this lung infections. And very briefly, NTM infections is regarded as the tuberculosis of the developed countries because we are having more NTM incidences in developed countries. Depending on the growth of mycobacteria, this is classified as 2 classes, So if you have rapidly growing bacteria infection, M. abscessus is problematic pathogens for your lung infections. And slowly growing bacteria, you have MAC infections. And like tuberculosis, this is very nasty pathogens to eradicate because these are environmental pathogens that gain resistance to most of everything, right? So you need to come up with a more effective, potent agent against this mycobacteria in general. For this market, you have lack of treatment. So for MAC, yes, you do have standard of care using a TB regimens, but these are not as active per se. The success rate is about 50%. mAb, you do not have standard of care, so you need to come up with a very good SOCs. If you have anti-infective and anti-inflammatory effect, you can also target bronchiectasis patients, so I think that will be very beneficial. And if you have broad activity against NTM and Pseudomonas, obviously, then you have very good coverage for the chronic lung infections. This is a market share. I will briefly mention it. So for MAC, you are taking these 3 oral regimens for over 12 months. And this causes significant systemic toxicity, and some patients cannot tolerate these treatments. And the key, I guess, the cornerstone of the drug is this macrolide antibiotics. And rifampin and ethambutol are basically just added to reduce the resistance of macrolides. So if you have resistance to macrolides, then you have basically no option. So you need to come up with a new agent. So the approved drug is Arikayce. This is an IV form of amikacin that's been repurposed to do the lung inhalations. And inhaled drugs are actually doing much better than the oral drugs in this field, as oral programs have been terminated for their systemic toxicity and whatnot. For mAb, you do not have effective agent. There's no SOC. So Arikayce is doing extension trial for the mAb approval, obviously. And there's omadacycline and another program in the early stage of the development. So we are doing the parallel investigation of the NTM, MAC, and mAb infections at the same time. And you need to have this optimal regimen for mAb. Otherwise, I don't think you're in a good game. So we are trying to optimize that as well at the earlier stage of the development. So this is the profile of our another candidate, AJ99, against NTM species. And because we have new mode of action, we have no cross-resistance to approved antibiotics. So even with the clarithromycin resistance, amikacin resistance, or depending on the MAC or mAb, we have potent antibacterial activity against a lot of different clinical strains, which is kind of neat. And like tuberculosis, NTM can reside in your macrophages, so you need to evaluate your compound's efficacy in macrophages. So clarithromycin is known to have actual very potent activity in these macrophages. And like our agent is also very potent in these macrophages. The recent articles are saying that maybe ethambutol and rifampin are not effective. They're just added to reduce clarithromycin. And this is kind of like exemplified in this diagram as well, as the 3 regimens that we use is comparable to the clarithromycin that you see there. Now, in order to increase patient compliance, obviously, you need to reduce treatment time or the frequency or the dosing regimen of your agent. So our agent actually has synergistic effect with clarithromycin. So when we do the 2 regimen as opposed to the 3 regimen, we have more antibacterial activity that's being seen. So targeting the patient compliance may be suitable for this development. And this is nicely illustrated in the in vivo model as well. So we do this progressive infection model. So we infect for 5 weeks, and treatment ensued for about 8 weeks. Our compound has been intranasally administered, and this shows activity in this NTM species. Not as great as clarithromycin, but importantly, because this is a combinatory regimen like tuberculosis, what we have to see is how well your compound does in terms of adding this regimens. So our agent is doing much better than that of the standard of care that we see today. And we see a lot of inflammation dampening of our agent. So despite the low reduction of CFU, you have more inflammation reduction of our agent. We are elucidating the mechanism of anti-inflammatory effect, but I do think that our agent has additional anti-inflammatory effect, just like macrolides that we see today. And this is nicely illustrated by lung H&E stainings, and the inflammations are remarkably reduced when we treat that with clarithromycin. And I won't go into much detail about Pseudomonas. This is another problem. So we are also targeting bronchiectasis patients. I think we can also target cystic fibrosis patients. For cystic fibrosis patients, this is the first line of treatments, and obviously, this Pseudomonas are gaining resistance to all of these antibiotics that you see. So, as I mentioned, we want to target inflammatory effect to reduce inflammations that we see for bronchiectasis and cystic fibrosis, and targeting both NTM and Pseudomonas. There's no effective drug available for it. And this is our in vivo model that shows our agent is active against Pseudomonas. This is in vivo. So we compare our agent with solution and suspension version of the molecule. Why suspension? Because one of our competitors is actually developing a suspension version of clofazimine as opposed to solution, because those are not soluble. And obviously, the solution is more effective against the suspension version of your drug product, in a sense. And our agent also has potent antibacterial activity against a very nasty, multi-drug resistant form of Pseudomonas. So I think that will probably address significant unmet needs in this chronic lung infection field. And I guess I can briefly show you that we have promising antibacterial activity derived from natural product, that we have done medicinal chemistry campaign on it, and doing preclinical and clinical development soon, and hopefully, have a very good antibiotic, for tomorrow's, address tomorrow's future. Thank you so much for your attention.